Hi, my name is Lisa Fritz. I'm a biological science technician working with USDA's Agricultural Research Service in Jackson, Tennessee. Today I'm going to tell you about developing soybeans in West Tennessee for improved resistance to Cyst Nematode. I work with Dr. Prakash Areli, and we're located at the West Tennessee Research and Education Center in Jackson. We conduct research both at the center in Jackson as well as at the Research and Education Center at Milan. This presentation will be organized into 3 sections, working backwards through the title. I'll start out with a little refresher and some pictures of Cyst Nematode. Then I'll talk about improved resistance and what I mean and where that comes from. And then I'll finish off by telling you about the soybeans we're developing here in West Tennessee. Soybean cyst nematode is a plant parasitic roundworm that infects the roots of the soybean plants. So you'll never see the eggs or the juveniles or the male worms without a microscope, but the female adult worm swells into a visible size when her body becomes a protective cyst that encases the eggs. And that's where it gets its name, soybean cyst nematode. So here you can see roots of a soybean plant, Hutchison, a variety you may be familiar with, and you can see these tiny little pale yellow dots on the root. So in this series of pictures, you can see the cysts much more clearly. These were taken under a microscope, and again, these are cysts formed on the roots of soybean Hutchison that were grown in our greenhouse, and you can really clearly see how the cysts are attached to and embedded in the root tissue. So how do you know if you have a problem with soybean cyst nematode in your fields? Well, the number one indication is yield loss. Each year, extension specialists and other professionals in the U.S. and Canada come up with disease loss estimates to determine the economic impact that diseases and other pests have on soybeans. The publication shown here from Plant Health Progress, published in 2017, contains summarized data for 28 U.S. states that grow soybeans, as well as Ontario, Canada. Over the duration of the survey, which was a 5-year period starting in 2010 and through 2014, soybean cyst nematode was estimated to have caused more than twice as much yield loss as any other disease. And when you look at the overall totals, the total estimated yield loss over that 5-year period due to all diseases was over 2.1 trillion bushels. But soybean cyst nematode accounted for about 617 million bushels of that loss, which is over a quarter of the total loss due to all diseases. If you'd like to know what the cyst nematode counts are for your own fields and you're located in Tennessee, you can find out that information for free by submitting a soil sample. Simply go to utcrops.com and then navigate to soybean and then to diseases and nematodes. Scroll down the list of documents and you'll find one entitled Nematode Sampling Guide and Submission Form 739. Simply open that up, print it out, and submit it along with your field soil samples. If you're located in another state, check with your county extension agent to see whether this service is available in your area. Next, I want to talk about improved resistance. When I use this term, or when I talk about a source of resistance or a new source of resistance, I'm talking about genetic resistance. So soybean plants can have resistance to soybean cyst nematode in the same way a person can have resistance to a disease, or a bacteria can have resistance to an antibiotic, or a plant such as a weed can have resistance to an herbicide. When we talk about improving resistance in a soybean, the source of resistance, where that genetic material comes from, is another soybean. Soybeans come in all kinds of shapes, colors, and sizes, and they are native to East Asia. The USDA soybean germplasm collection contains over 20,000 accessions of soybeans from all over the world. A curator may collect material in the wild, or it could be submitted, but each entry is cataloged and given a PI or plant introduction number. The many uses of the collection is that it enables researchers like us to search through diverse soybeans to find ones with new, unique genes for Cyst Nematode that are not already present in commercial varieties in this country. Of the thousands of lines in the collection, there are only 3 plant introductions that are providing the SCN resistance in the soybean varieties you can buy on the market today. These 3 lines are PI 88788, Peking, and PI 437654, or Hartwig. PI 88788 was found in China and introduced into the collection in 1930. Varieties with PI 88788 resistance dominate the market. Because the PI 88788 source has been incorporated into so many different soybean cultivars over so many years, prolonged use has led to shifts in nematode populations. This means that the PI 88788 source of resistance is no longer a great match to the populations of Cyst Nematode that we have in Tennessee. You probably know that when we talk about Cyst Nematode, we talk about different races. And races describe the relationship between a Cyst Nematode population and the different sources of resistance. So PI 88788 is characterized as having resistance to races 3 and 14, but the populations of nematodes we're finding in Tennessee now are predominantly races 2 and 5. In the bottom right corner of the slide, you'll see that I've included information on how you can identify the 88788 source of resistance when you're looking at a soybean to purchase. So you may see the source named as PI 88788, or you may see the resistance. If you see a soybean bag labeled with resistance to races 3 and 14, or resistant to race 3, moderately resistant to race 14, you can be sure that that soybean was developed using the PI 88788 SCN resistance source. Next up is soybean Peking, which has a PI number of 548402. This line was brought to the U.S. from China in 1954. Peking was used to develop a lot of southern cultivars, but a lot of those early cultivars didn't get all of the resistance genes. So while Peking itself is resistant to SCN races 1, 3, and 5, many of the lines developed from Peking are only resistant to races 1 and 3. For the maturity groups we grow in Tennessee, you won't find very many cultivars with the Peking source of resistance, but there are a few out there. So if you're looking on a company website or at a seed bag and you see Peking listed as a source of resistance, or you see resistance to SCN races 1 and 3, that's an indication that you have the Peking source of resistance and that is different from the PI 88788 source of resistance. This is important because if you have an SCN-infested field, some of your best management practices are rotation. Not only rotation of soybean with a non-host crop, like corn or sorghum, but also rotation of resistant varieties. Best yet, rotating resistant varieties that have genetic material from different sources of resistance. The third source of resistance available to you today comes from this soybean, PI 437654. It came to the U.S. from China in 1979. Now, PI 437654 was crossed to a line derived from Peking to create another soybean called Hartwig. And a lot of the lines that have been developed from the 437654 source were actually developed from Hartwig. So these two soybeans, these two names, are not identical or interchangeable, but they do have the same resistance. They are resistant to races 1, 2, 3, 5, and 14 of Cyst Nematode. And you'll remember from earlier that races 2 and 5 are those most commonly found in fields in Tennessee. The availability of cultivars developed from this resistant source is not particularly widespread, but I'll give you some tips to identify it when you see it. So the source could be either listed as Hartwig or as PI 437654. Again, the resistance in most lines comes from 437654 through Hartwig. You may see the resistance to SCN races listed in any number of different ways, but one thing they'll have in common is they list race 2. Of the 3 sources we've talked about, only 437654, or Hartwig, has resistance to race 2. Dr. Relly joined the program in 2001, and I followed in 2002. The 5 soybeans that you're seeing in the slides are all lines we've released from 2005 to 2017 that have the Hartwig source of resistance. One of the main goals of our breeding program in Jackson is to improve resistance to Cyst Nematode by finding new genetic sources. This will give you more options to rotate and plant in your field, which will help suppress the nematode populations and maintain the viability of soybean as a crop. Last year, in 2019, we released 3 lines that were something new. They combined the resistance of Hartwig with a different genetic source called PI 567-516C. These 3 lines, called JTN 5316, JTN 5416, and JTN 5516, have been released and distributed as breeding material for use by other breeding programs. Looking ahead, we have a lot of exciting things in the pipeline. We're working on combining the Hartwig source of resistance with resistance to the fungal pathogens that cause charcoal rot of soybean and frog eye leaf spot, 2 very economically important diseases. This project is in combination with Dr. Alamu Mengistu and Jason Deffenbaugh, who have another talk in this field day. We're also working on developing soybean lines from new source PI 437655, as well as new source PI 494182. Both of these are genetically different from the sources that are already available to you. We're also working on combining some of these sources together. So new source PI 567-516C, which was in the new lines that we released in 2019, we're also working on combining that with some of these other new sources, like PI 494182. Now that we've covered the different resistance sources available for Cyst Nematode, both currently and what we hope to develop in the future, I want to change gears and show you how we accomplish our research. We have a field, lab, and greenhouse component to our research, and I'll start out by showing you a few slides from planting. We plant anywhere from 500 to 1,500 different varieties each year, and we package our seed in envelopes and plant them with a cone planter. These seed have been packaged for 12-foot rows, so there's about 120 seeds per envelope. Now you may have noticed in the planter picture that the field behind us had been tilled. This was in Jackson in 2019. We do normally plant in a no-till environment, and especially at the Milan station. We plant 30-inch rows, and the main reason for this is that we need to be able to walk between the rows to take extensive notes on all the different lines throughout the season. We take notes on flower color, primarily to see if the row is uniform or not, as well as any diseases that occur throughout the season, height, lodging, shattering, green stem, and maturity. While you're probably used to harvesting into something that looks like this, well, our harvest process looks a little different. We harvest each short plot into small bags that are brought back to our seed room, which is like a mini warehouse. The average harvest per plot is between 1/2 and about 6 pounds. Over the course of the winter, seed will be analyzed in several different ways. Lines that do well in Jackson and Milan may go on to multi-state regional trials like the USDA Uniform Soybean Tests or to state variety trials. Our greenhouses are an essential part of our research. This is the primary place where we evaluate the lines we're developing to see if they're actually resistant to soybean cyst nematode. The seed are planted in the greenhouse and inoculated with the eggs of the cyst nematode. After about 30 days, which is one reproductive cycle, the plants are pulled up and the cysts that have attached to the roots of each plant are counted. If you look closely, you may see that some of these plants are PI 88788, PI 437654, and Peking are SCN-resistant sources. These are part of a set of indicator lines, which help ensure that our greenhouse screening data is consistent and reproducible. In our genetics lab, we compare the DNA of the lines we're developing to the resistant source to try to determine which lines are likely to have cyst nematode resistance and should be kept, and which ones aren't likely to have resistance and can be discarded. This method is called marker-assisted selection, and it's a way we can narrow down the number of lines going into the greenhouse for the more labor-intensive screening. And now for the take-home message. Do you know where your fields stand with cyst nematode? If not, get them tested. It's free in Tennessee. When it comes to the seed that you plant in your fields, rotation is key to managing cyst nematode. Be sure to pay attention to the SCN-resistant source for the seed you're buying and mix varieties up as much as possible. Thanks for listening. I hope you have a great harvest season, and I hope to see you at No-Till Field Day in 2022. Take care.